Okay, so welcome back to this next video in which we are discussing depression and the antidepressants. Okay, right, so we are in the process of discussing serotonergic neurons. So we have discussed how serotonin, or 5-hydroxytryptamine, is synthesized in axon terminals from the amino acid tryptophan in this two-stage process. Firstly, you add an alcohol group onto the 5th carbon of the indole ring of the tryptophan amino acid to produce 5-hydroxytryptophan, or 5-HTP here, okay, and then you decarboxylate the 5-hydroxytryptophan to produce 5-hydroxytryptamine, and that conversion is done by the L-aromatic amino acid decarboxylase enzyme. Okay, right, then the 5-hydroxytryptamine that's in the cytoplasm of the axon terminal is going to be loaded into synaptic vesicles by the vesicular monoamine transporter 2, okay, which is going to move one serotonin molecule in for 2 protons moving out. Now, protons have a very high concentration in the lumen of the vesicle, and this is because they are pumped into the lumen by a proton pump on the membrane, well, in the membrane of the synaptic vesicle that is a primary active transporter and is using ATP hydrolysis to move the protons into the lumen of that vesicle. Okay, VMAT2 in contrast is a secondary active transporter. It uses the energy that is released when the protons leave the synaptic vesicle to move the serotonin into the synaptic vesicle. Okay, the other fate for serotonin is that it could be broken down, okay, by an enzyme that is is attached to the outer membrane of mitochondria. Okay, so let's draw this here. So in the axon terminal of all neurons, you have a lot of mitochondria. Okay, and I'll draw a little picture of a mitochondria here. So remember, mitochondria have 2 membranes. They have an inner membrane and an outer membrane, shown here. Okay, so this is supposed to be a mitochondrion. So this here, this is the outer mitochondrial membrane, and both of these membranes are full lipid bilayers. Okay, so this is the outer mitochondrial membrane, and then inside the outer mitochondrial membrane, you then have the inner mitochondrial membrane here. Okay, and you then have a space in between the outer membrane and the inner membrane. Okay, and that's called the intermembrane space. Okay, now the inner membrane, you'll see that it's not just in a simple oval shape. Instead, it has these invaginations inwards. Okay, and these are known as cristae. Okay, so these are little invaginations inwards. Those are called cristae. Okay, and those increase the surface area of the mitochondria. Of the mitochondrial inner membrane, and remember, it's the mitochondrial inner membrane that is the site of the electron transport chain. Okay, so you want it to be as large a surface area as possible. Now, there are enzymes that are attached to the outer mitochondrial membrane, okay, and these enzymes are capable of breaking down 5-hydroxytryptamine. So let me draw this on the other side. Okay, we actually haven't got that much more room here, but we have a little bit more room. So let's say that these 2 lines here, okay, represent the 2 layers of the lipid bilayer of the outer membrane, okay, of the mitochondrion. Okay, now let's say the top of this membrane is the cytoplasm, okay, and then the bottom of this membrane is the intermembrane space. Okay, so this will go towards the inner membrane of the mitochondrion. Okay, so this is the intermembrane space down here. Okay, so I'm now going to show this enzyme that's going to be attached to the outer membrane, and it's going to have most of its bulk in the cytoplasm. So the portion that's actually going to break down serotonin is going to be in the cytoplasm, but then it has a membrane spanning alpha helix which spans the outer membrane, and it's going to have its C-terminal tail in the intermembrane space, whilst its amino terminus will be cytoplasmically located. Okay, so this enzyme then is implanted into the outer membrane of the mitochondrion by a single membrane spanning alpha helix here, but the main bulk of it is floating around in the cytoplasm here. Okay, so what is this enzyme called? Well, it's called monoamine monoamine oxidase. Okay, and it breaks down all of the monoamines. It breaks down dopamine, serotonin, and noradrenaline. So we're going to see a lot more of this. Okay, and for short, monoamine oxidase is often abbreviated to MAO for short. M for mono, A for amine, and then O for oxidase. Now there are actually two different types of monoamine oxidase. Okay, there is monoamine oxidase A. Okay, and there is also monoamine oxidase B. Okay, now monoamine oxidase A is very good at breaking down serotonin. Okay, serotonin is the monoamine it loves to work on. Okay, it will break down noradrenaline and dopamine as well, but its preference is for 5-HT. Monoamine oxidase B, again, it will break down all three monoamines that we've mentioned. Okay, but its particular favourite is dopamine. Okay, and for short, dopamine is often abbreviated as DA, D for dope, and then A for amine. Okay, right. So, if we're talking about serotonin, then we're going to be particularly interested in monoamine oxidase A, but monoamine oxidase B will break down serotonin, it's just, it much prefers dopamine. Okay, so let's put this onto our picture here. Okay, so on the outer membrane of the mitochondrion then, here is our monoamine oxidase enzyme, and it's either monoamine oxidase A or monoamine oxidase B. Okay, and any 5-HT that comes into contact with the monoamine oxidase enzymes will be broken down. So not all of the serotonin that you produce will actually end up in the synaptic vesicle. Some of it will be broken down by monoamine oxidase enzymes. Okay, so that might seem slightly wasteful, but it's the way it's done. Okay, you produce lots of serotonin, some of it will end up being packaged into the synaptic vesicles, some of it will just be made to be destroyed by monoamine oxidase enzymes. Okay, it ensures that you don't produce too much, basically. Okay, so then what's going to happen is when an action potential arrives, that will cause the release of the serotonin. Okay, so the synaptic vesicles will fuse with the presynaptic membrane here, releasing the serotonin into the extracellular fluid. Okay, once it's been released, there are transporters, okay, on the presynaptic membrane here, which will then transport the serotonin back into the cytoplasm of the axon terminal. And then once it's back in the cytoplasm of the axon terminal, it will either be reloaded into synaptic vesicles or it will be broken down by the monoamine oxidase enzymes. Okay, so now what we're going to study is this transporter, because this is very, very important. Okay, this is going to be the target for many of the antidepressant drugs. Okay, this is what's known as the serotonin reuptake transporter, and again, it is within the family of solute carrier transporters that we saw earlier. It's not within the solute carrier transporter family 18, it's actually within the solute carrier transporter family 6. Okay, but it's still within that massive great superfamily of transporters. So let's study this transporter now in more detail. So, firstly, for short, the serotonin reuptake transporter is often abbreviated to SERT, okay? SE for serotonin, R for reuptake, and then T for transporter. Okay, and it's in the family of solute carrier proteins, family number 6. Okay, so it's in the SLC6 family. Now, in the SLC6 family, you have 20 members. Okay, so it's not a small family of solute carrier transporters, it's a big family, bigger than SLC18 was anyway. Okay, and these 20 members are called SLC6A1, SLC6A2, SLC6A3, SLC6A4, all the way up to SLC6A20. Okay, so it's a nice, simple naming system for the SLC6 family members. Okay, right. Now, many of the SLC6 family of transporters have nothing to do with serotonin. They transport amino acids. Okay, some of them even transport the amino acid GABA. Okay, the ones that we're going to be particularly interested in is a subfamily of the SLC6 proteins, known as the monoamine transporters. Okay, so just to put this in perspective, there is the massive great family of SLC transporters, okay, solute carrier transporters. Within that massive superfamily, there is a family known as the SLC6 family of transporters. In that family there's 20 members, and we're now dividing this family up even further into subfamilies, and one of the subfamilies that contains 3 members is the monoamine transporters. Okay, and obviously we're going to be interested in these, and SERT is going to be one of these monoamine transporters. So the 3 monoamine transporters are SLC6A2, okay, SLC6A3, and SLC6A4. Okay, now those are their official names. They all have other names that are a bit more in common use. Okay, so SLC6A2 is the transporter for dopamine. So it's not just a property of serotonergic neurons that you have a transporter which reuptakes the neurotransmitter that you've released. Many neurons do that. The dopaminergic neurons and the noradrenergic neurons also do that. So the DAT is the dopamine, so DA is for dopamine, okay, and then the T is for transporter. Okay, so this is on dopaminergic neurons, and it reuptakes dopamine after you have released the dopamine into the extracellular fluid. Okay, so that's SLC6A2. Okay, SLC6A3 is also known as the norepinephrine transporter, or for short, it's known as NET. Okay, so N is for nor, E is for epinephrine. Okay, and then T is for transporter. So this is when it becomes useful to know that noradrenaline is also called norepinephrine. Okay, in understanding why the why the transporter for noradrenaline is called the NET. Okay, right. So this is on noradrenergic neurons and will reuptake noradrenaline from the extracellular fluid into the axon terminal cytoplasm. Okay, and its official name is SLC6A3 there. Okay, right. And that leaves finally SLC6A4 is the serotonin reuptake transporter, or SERT. Okay, so this is the one we're interested in at the moment, but we will come on to the norepinephrine transporter further later on. Okay, so let me just describe to you the structure and the function of this transporter then. So, they have very similar structures to the SLC18 family members, so they have very similar structure to VMAT transporters. Okay, they again have their amino terminus on the cytoplasmic side. So if this is the membrane, let's say this is now the cell membrane rather than the membrane of the synaptic vesicle. Okay, so let's say the bottom is the cytoplasm once again, and then the top this time is the extracellular fluid, and for short I'll abbreviate extracellular fluid as ECF. Okay, then following the amino terminus, this line is representing the polypeptide, so it's representing a polymer of amino acids. Okay, so it represents amino acid after amino acid after amino acid, and basically what's going to happen is the polypeptide is going to straddle the membrane multiple times. Okay, and again it's going to be 12 times, just like was the case in the SLC12 family members, sorry, 18 family members rather than 12 family members. Okay, so here are the 12 membrane-spanning alpha helices, and then you'll finish with the C terminus, the carboxylic acid terminus, in the cytoplasm as well. Okay, right, so here are 12 membrane-spanning alpha helices. Now, the first 10 of these, okay, so I'll colour these in pink here. Okay, these first 10 are going to be involved in actually forming the portion that's going to be involved in transporting the serotonin from the extracellular fluid into the cytoplasm, whereas the final 2 sort of sit on their own, okay, and are thought to maybe be involved in the dimerization of 2 of these proteins together. Okay, so this structure, we're thinking of it in terms of the structure of the serotonin reuptake transporter, but it's the same for the dopamine transporter and the norepinephrine transporter as well. Okay, so these final two helices, they sit apart from the other 10, okay, and we think they might be involved in binding two of the transporters together, basically, okay, although you don't need two of the proteins to bind together in order to have a functional transporter. On its own, this protein is a functional transporter for serotonin. So, I'm now going to draw this protein in a different way, okay, so I'm now going to draw it like this, which is a functionally more meaningful picture. Okay, so all of these 10 portions here, all of these 10 membrane-spanning alpha helices are now being represented by this. Okay, so they're all aggregating together, and they're going to form the portion that's actually involved in transporting serotonin from the extracellular fluid side to the cytoplasmic side. Okay, then you have the final two membrane-spanning alpha helices here, and then the C-terminal here. Okay, and we'll colour in those final two portions in green. Okay, right. Now currently, this transporter, and we're imagining that it's the serotonin reuptake transporter, but you could imagine that this is also the norepinephrine transporter, because that's the one we're about to need when we move on to noradrenergic neurons. Okay, currently, this is in the outwardly facing state. Okay, so transporters have two states. This is a running theme for all transporters. Okay, they have one state which faces one side of the membrane. In this case, we're in the cell membrane, so we've got an outwardly facing state which is facing the outside world. Okay, and then we'll have another state which will face the opposite side of the membrane, in this case the cytoplasm, and that will be called the inwardly facing state. So we start off in the outwardly facing state in the case of both the serotonin reuptake transporter and the norepinephrine transporter, and of course the dopamine transporter as well, but we're not interested in that one. Okay, so then what's going to happen is you'll have a serotonin molecule in the extracellular fluid. This will come and bind to this little binding pocket that I've drawn here. Okay, along with two other things, okay? So once again, these transporters, the dopamine transporter, the norepinephrine transporter, and the serotonin reuptake transporter, they are not active transporters. Well, sorry, they are active transporters, but they're not primary active transporters. Okay, they do not couple the reuptake of serotonin, noradrenaline, and dopamine to the hydrolysis of ATP. Instead, they couple it to the movement of ions down their gradient. Okay, so the ions that are going to move are sodium and chloride. Okay, so remember there is a fantastic sodium gradient across the cell. Okay, the concentration of sodium extracellularly is around 145 millimolar, whereas it's 12 millimolar intracellularly. Okay, so there's a fantastic gradient driving the movement of sodium into the cell. Okay, so sodium will bind here, and a chloride anion will also bind here. Okay, chloride generally also has a concentration gradient favouring its movement into the cell. Okay, and then what's going to happen is once you've got these three things bound, a 5-HT molecule, a sodium ion, and a chloride anion, that will cause the transporter to change in conformation now. Okay, and it will change into what's known as the inwardly facing state. Okay, so a huge, great conformational change will happen, and now the molecules will be moved from the outside to the inside. Okay, so let me just draw the full molecule here. So here is the cell membrane, here is the C-terminus, here's the amino terminus. Okay, right, so let's colour these bits in again here. Okay, I'll just move this up. Right, so here, the main bulk of the transporter, once again, I'll show in vivid pink here. Okay, then we'll have the two accessory bits which are thought to be involved in the dimerization of two transporters together. Okay, and once we're in this inwardly facing state now, when the binding pocket is facing the cytoplasm, okay, now what will happen is the binding pocket loses its affinity for those three molecules that are bound. So it loses its affinity for 5-hydroxytryptamine, it loses its affinity for the sodium ion, and it loses its affinity for the chloride anion. Okay, so all of them will be released onto the inside, and now once they have been released, now the transporter will change back in conformation to the inwardly facing state, and then it will have a high affinity again for the 5-hydroxytryptamine, the sodium and the chloride. So to summarize, when you're in the outwardly facing state, you have a high affinity for the solute molecule, for these three molecules, the substrate, the sodium and the chloride. Okay? When they bind, they cause you to change conformation, and you move into a conformation where you have low affinity for those molecules now, but by moving into this conformation you have moved them inadvertently across the membrane, and then when you lose your affinity and let go of them, they're released into the cytoplasm. Then what will happen is, now that you no longer have them bound, you change back to original conformation, and then you're in a high affinity state again for them. Okay, so that's the general principle of how transporters work. Okay, so once again, the serotonin reuptake transporter is a secondary active transporter rather than a primary active transporter. Okay, right. So that now concludes our discussion of serotonergic neurons. Okay, now what I want to do is do the same thing now for norepinephrine. Okay, because the monoamine hypothesis, the two main monoamines are serotonin and noradrenaline, not just serotonin, okay, noradrenaline as well. And many of the antidepressants work by raising noradrenaline levels as well as serotonin levels. Okay, so it's important that we look at norepinephrinergic neurons as well as just serotonergic neurons. Okay, right. So, shall I do it here? Okay, so I won't draw a massive great axon terminal again. I'll just put a small axon terminal this time. So we're now studying noradrenergic neurons. Okay, written like so. Noradrenergic, or you can, if you like, write it norepinephrinergic, but that's a bit more of a mouthful. Okay, so this is a noradrenergic neuron. Okay, right. And what we want to study is, firstly, once again, how do you synthesize noradrenaline? Because again, it's synthesized from an amino acid precursor. Okay, and in this process we'll see also the production of dopamine, because dopamine is a precursor to noradrenaline. Okay, then we'll discuss how it's packed into vesicles. It's exactly the same as how it's packed into vesicles for serotonin. Okay, and then we'll discuss monoamine oxidase degradation and also the reuptake by the norepinephrine transporter. Okay, so synthesis then. So noradrenaline starts with tyrosine. Okay, starts with the amino acid tyrosine rather than tryptophan. So let's show the structure of tyrosine. So once again, we'll start with the core amino acid structure. So here's the amino group. Okay, here's the alpha carbon with a hydrogen coming off it, and here is the carboxylic acid group here. Okay, like so. And then the R group of tyrosine is a methylene group. Okay, then with a benzene ring coming off this methylene group. Okay, so here is my benzene ring, like so. And then off the benzene ring you then have an alcohol group. Okay, so this is the structure of the amino acid tyrosine, and this is what we're going to start off with in the synthesis of noradrenaline. Okay, so the first thing we're going to do is we're going to add an additional alcohol group onto tyrosine. Okay, so you're going to start with tyrosine, and you're firstly going to add an alcohol group on. Now I'm not going to draw a whole new picture just for this time, okay? I'm just going to change this picture here, okay? So take a picture of what tyrosine is in your head now. We're going to change it. We're going to add on an alcohol group on here. Okay, so we're going to add an additional alcohol group onto the benzene ring within tyrosine. Okay, now there is a special name for a benzene ring, like so. Okay, so I'll colour this in. So here is the benzene ring with two alcohol groups coming off. Okay, so with these two alcohol groups coming off, there is a special name for that in chemistry. Okay, a benzene ring with two alcohol groups coming off is known as a catechol ring. Okay, so we have now created a catechol ring, and this catechol ring is going to be present within both dopamine and noradrenaline. Okay, and this is the origin of dopamine and noradrenaline, and also adrenaline itself, although we won't quite go along the synthesis pathway to adrenaline. Okay, it's the origin of them being known as catecholamines. Okay, because they're going to have a catechol ring within their structure, and they're also going to have an amine group. Okay, so that's a nice little correlate of that. Okay, so the enzyme that does this then is known as tyrosine hydroxylase. Okay, and again, it is within the cytoplasm of the axon terminal. Okay, so tyrosine gets converted to, oh, I haven't even told you what this molecule is yet. Okay, so this is a famous molecule. The instant I tell you the name of this, you will probably recognize it. Okay, this is known as dopa. Okay, now what does dopa actually stand for? Well, it stands for dihydroxy, so the D is for di. Okay, the O is kind of the oxy in hydroxy. Okay, and then the PA is for phenylalanine. Okay, so firstly let me tell you about the amino acid phenylalanine. Okay, so basically phenylalanine is alanine with a phenyl group on it. A phenyl group is just a name for a benzene ring. So in fact, if you take this structure here and you take the two alcohol groups off, okay, so imagine taking this off and taking this off, that is phenylalanine. It's another protenergic amino acid. It's tyrosine without the alcohol group, basically. Okay, so this name makes sense because this is phenylalanine with two alcohol groups added on, basically, to create the catechol ring that we have. Okay, so that's the origin of the name dopa. Now, why is dopa so famous? Well, it's because the very famous anti-Parkinson's drug, or the very famous treatment for Parkinson's disease, is L-dopa. Okay, and all that's referring to is the fact that this, the chiral center here, the enantiomer that we're using is the L enantiomer, which of course it was the one we were using, okay, all along. I never stated that explicitly, but in cells you always use the L enantiomer, not the D enantiomer. Okay, so L-dopa, this is L-dopa is the reality, but that's the what's given in Parkinson's disease, and the reason it's given is to increase the synthesis of dopamine, which is what you have too little of in Parkinson's disease and causes the motor problems. Okay, right, so next what we're going to do is we're going to remove the carboxylic acid group from dopa to convert it into dopamine. Okay, so next up you're going to convert dopa into dopamine by removing this carboxylic acid group here. Okay, so just like in the case of tryptophan, you're going to remove that carboxylic acid group and replace it with a hydrogen. Okay, and I think I will redraw this now. Now, do I dare try and squash it in there? Okay, I don't think I'm going to try. Oh, well, I'll try, okay. So here's the amino group here. Okay, here's the alpha carbon. Okay, and now it's had that carboxylic acid group lost, and it's got a hydrogen there instead. Okay, then we've got the methylene group here. Okay, then we've got the benzene ring with the two alcohol groups coming off, which create this, well, which turn this into a catechol ring. Okay, so that then is the structure of dopamine. Okay, so that's the neurotransmitter that we've been hearing so much about. Okay, so that's one of the monoamines, and now we can see why this is a monoamine, because it has a single amino group. And I should have probably actually said that with serotonin. Okay, let me get my serotonin structure back again. It's got a single amino group, that's why it's a monoamine. Okay, right, so now we're going to convert dopamine into noradrenaline. But before I tell you about that, let me just tell you about the fact that, the enzyme that catalyzes the conversion of dopa to dopamine. Okay, so the enzyme which catalyzes this is again the L-aromatic amino acid decarboxylase, the LAD enzyme. Okay, right. Now what happens is dopamine is transported into the synaptic vesicles by the vesicular monoamine transporter 2, and then in the synaptic vesicles it is then converted into noradrenaline. Okay, so dopamine is going to go in there, and then the next enzyme that's going to convert dopamine into noradrenaline is actually located within the synaptic vesicles. Okay, so so how do you convert dopamine into noradrenaline? Well, now I'm just going to have to draw out the picture of dopamine again. So here's the amino group. Okay, here's the alpha carbon with 2 hydrogens coming off it now. Here's the methylene group. Okay, and here is the benzene ring with 2 alcohol groups coming off it, like so. Okay, right. So to convert dopamine, as seen here, into noradrenaline, what you do is you remove this hydrogen here, okay, and you replace it with an alcohol group, okay. Therefore, the enzyme which converts dopamine into noradrenaline is known as dopamine hydroxidase. And as I say, this enzyme is not floating around in the free cytoplasm of the axon terminal. Instead, it is within the synaptic vesicles. Okay, so the dopamine is first transported into the synaptic vesicles, and then it's converted into noradrenaline. Okay, now, that doesn't mean that VMAT2 can't actually transport noradrenaline from the cytoplasm into the synaptic vesicles. It can, and we'll see why it needs to be able to in a moment. Okay, so here is the alpha carbon with 2 hydrogens coming off it, and now this carbon here is now going to have an alcohol group coming off here. Okay, and then we're going to have the catechol ring here, so the benzene ring with the 2 alcohol groups coming off. Okay, right, and this now is the structure of noradrenaline or norepinephrine, depending on your preferential name. Okay, right, so this is noradrenaline. Right, so now we have got noradrenaline. Now, what I would just like to mention is the fact that this too is susceptible to being degraded by monoamine oxidase enzymes. Okay, so remember, in this synthesis pathway we synthesize dopamine in the cytoplasm, and then the dopamine is pumped into the synaptic vesicles. However, dopamine can be broken down by monoamine oxidase enzymes, particularly monoamine oxidase B enzymes, so not all of the dopamine will make it into the synaptic vesicles. Okay, then the dopamine that does make it into the synaptic vesicles will be converted to noradrenaline. Okay, now, if we draw our axon terminal here, the noradrenaline will be released from the axon terminal when the action potential arrives at the axon terminal, and it will go into the extracellular fluid, and then it's going to be reuptaken back from the extracellular fluid into the cytoplasm by the norepinephrine transporter. Now, the norepinephrine transporter is very, very similar to the serotonin reuptake transporter. Once again, it is the SLC6A3 protein. Its structure is exactly the same as the serotonin reuptake transporter. Okay, so it too looks exactly like this. Again, it sits in the plasma membrane like so. It starts in this outwardly facing state. A noradrenaline molecule binds to it. So does a sodium ion, and so does a chloride anion. Okay, then it changes conformation to the inwardly facing state, which has low affinity for all of these molecules, so they are released into the cytoplasm. Once it's lost them, it returns to the outwardly facing state, which then again has the high affinity for those 3 molecules. Okay, and that's how it's moving noradrenaline into the cytoplasm of the cell from the extracellular fluid. Okay, now, what is the point of reuptaking the noradrenaline? Well, firstly, it is to cancel the signal, but also you want to put the noradrenaline back into the synaptic vesicles, and that's why VMAT2 needs to be able to transport noradrenaline as well as just dopamine. Okay, so VMAT2 will then move the noradrenaline back into the synaptic vesicle, okay, or the noradrenaline in the cytoplasm will be degraded by either monoamine oxidase A enzymes or monoamine oxidase B enzymes, which are, remember, attached to the outer membrane of the mitochondria. Okay, right. So, now let me discuss the monoamine hypothesis in more detail. Particularly, I want to discuss the main two drugs which gave rise to the monoamine hypothesis. So the first drug that gave rise to the monoamine hypothesis was a very famous drug known as reserpine. Okay, so reserpine was being used as a antihypertensive drug. Okay, now the way it worked was that it bound to the vesicular monoamine transporter, okay, and stopped it from functioning. Okay, so it stops the function of the vesicular monoamine transporter, and thereby it stopped the loading of dopamine and also serotonin and also noradrenaline into the synaptic vesicles, and therefore all of your neurons are going to stop having noradrenaline, dopamine, and serotonin in their synaptic vesicles. Now, why was that helpful for hypertension? Well, basically, you have noradrenergic neurons innervating the smooth muscle cells that surround your arteries. Okay, so if I have a blood vessel here, okay, there is smooth muscle cells surrounding the blood vessels. Okay, so I'll show one here. Okay. Now, these smooth muscle cells are innervated by noradrenergic neurons, okay? So this neuron will release noradrenaline, okay? And noradrenaline causes those smooth muscle cells to contract, okay, and constrict the blood vessels, okay? So if you stop all neurons in the body from being able to store noradrenaline in synaptic vesicles, then all of these neurons that innervate the smooth muscle cells that surround blood vessels are going to be depleted of noradrenaline. Okay, so they're no longer going to be releasing noradrenaline onto these smooth muscle cells, and they're no longer going to be causing them to contract. So all of the smooth muscle cells that surround your blood vessels stop contracting, and they all relax, and all your blood vessels dilate therefore. Okay, and that means that your blood is now in a higher volume, and therefore your blood pressure goes down. Okay, so it was a fantastic antihypertensive drug. It really was fantastic at bringing blood pressure down. The problem is, it caused severe depression, okay, in a lot of people who took it, okay, so I think it was something like 20% of the people who took it, it caused depression, okay, and some of them committed suicide whilst on it, so it was a terrible, terrible drug, and it's no longer used as a antihypertensive. Okay, in fact, it's no longer used at all. So, why did it cause the severe depression? Well, what we knew that it did is that it depleted dopamine in the brain, it depleted serotonin in the brain, and it depleted noradrenaline. It effectively turned all of the monoamine systems that we've spent so long now describing off. Okay, and that caused severe depression. Okay, so that was the first drug that offered some favour towards this monoamine hypothesis. In fact, this was what spawned the monoamine hypothesis. Now, after that came along another convincing argument, okay, in the form of the drug iproniazid. Okay, now iproniazid is not a particularly nice drug either. Okay, it used to be used to treat tuberculosis infections. Okay, many drugs ending in iazid are used to treat tuberculosis. So isoniazid is another example. Okay, in fact it's niazid, the ending that deems that you are used against tuberculosis. I think specifically they inhibit the synthesis of mycolic acid or something, but don't hold me on that. Okay, so iproniazid was this anti-tuberculosis drug, and it was found that it was actually effective as an antidepressant, and this was the first antidepressant that anyone had ever found. Okay, so it was the first useful antidepressant. It actually produced antidepressant effects. Okay, now how was it found to produce antidepressant effects? Well, in actual fact, iproniazid turns out to inhibit monoamine oxidase enzymes. Okay, it inhibits both monoamine oxidase enzymes. So it inhibits monoamine oxidase A enzymes and also monoamine oxidase B enzymes. Now remember what those enzymes were. They were the enzymes sitting attached to the outer membrane of the mitochondria. Okay, and you've got lots of mitochondria in the axon terminal. Okay, it needs a lot of energy. Okay, and these enzymes broke down monoamines, so they broke down dopamine, noradrenaline, serotonin. Okay, so if you inhibit those enzymes, then the noradrenaline, the serotonin, and the dopamine won't get broken down. You'll end up with vesicles utterly stuffed full of the neurotransmitter, and therefore you'll end up with higher levels in the brain. Okay, ta-da! You've got an antidepressant which raises the monoamines, and you've got a horrible pro-depressant which decreases the monoamines. And this was the beginning of the monoamine hypothesis. Okay, and we're now going to see that all the other antidepressants work in the same way as ipronizid. They raise monoamines. Okay, however, as I said earlier, the big problem with this theory is that antidepressants do this after you take one pill. Okay, they raise serotonin, dopamine, and noradrenaline levels in the brain, but they don't produce antidepressant effects after just taking one pill. You have to take them for weeks, okay, before you see any noticeable effect. Okay, why is that? Okay, what the general point of view is now is that the antidepressants work to increase the monoamine levels, particularly serotonin and noradrenaline are important. These cause some sort of remodeling of the brain, okay, maybe in the anterior cingulate cortex, if that's the place which goes wrong, which I think is the biggest lead that we have at the moment. That's the most likely area to be the cause of depression, okay, and then that heals the depression. Okay, so by raising monoamines, you help the brain to heal what the real pathology is, I think, is the general viewpoint of how these drugs work. Okay, right, and with that sort of stage-setting announcement, I will leave this video here now, and in the next video we will see the antidepressant drugs. Now this shouldn't take too long because we've actually seen all of the targets for the antidepressant drugs now, so all I have to do is now list a name of, list a list of drug names basically.